So very good morning everyone. In this video, I am going to discuss about first unit of RT550, that is a drug discovery and development. In this video, I am just giving you an overview about drug discovery and development process. So drug discovery and development process, as maybe many of you knows, it's very time consuming as well as a lot of money is involved in this process. So many pharmaceutical companies, particularly the pharmaceutical company of developing countries, are not investing their time and money in this process. But I personally feel money is not the only reasons, because big generic companies have sufficient amount of money that they can put in this process. But I feel personally the reason might be the attrition rate. Attrition rate means failure rate. Failure rate in drug discovery and development is very high. If suppose you have identified in early phases of drug development 10,000 molecules, out of 10,000 you are not sure about whether you are able to put one molecule out of 10,000 into the market or not. So attrition rate is very high, so that's why many companies are not putting. But yes, there are innovative companies like Pfizer, Johnson and Johnson, Novartis, who are working in drug discovery and development. So what actually the different phases in drug discovery and development? So first phase in drug discovery, you have to identify a target. So first one is target identification. First we have to identify a target. And how we can identify a target? We have to first understand about pathogenesis of that particular disease in much more detail. If we are understanding in much more deeply about the pathogenesis, we can identify a specific target for that particular disease, and then we can target these targets. So first step is you have to identify a target, and target could be anything. Target could be your protein, target could be your DNA, RNA, target could be the enzymes. But in infectious diseases, target could be host target or microbial target. If I give you examples of recent pandemic situations, in coronas we can target the host enzymes, because virus is enter inside the cells and use the host machinery for the replication or maturation. So either we can target the host enzymes, which helps in the replication or maturations of that particular enzymes, or we can target the specific microbial targets like spike protein. Spike protein is not present in the host, so many research is going on or to target the spike proteins as well as for the host target. So target identifications, first step, and after that you have to validate that target. Suppose you have identified 5 6 target or 7 8 target. So out of these targets, which one is the best target for that particular disease? Yeah, and by targeting that one, whether you have to need a agonist, antagonist, or partial agonist or partial antagonist for that target. And after targeting that particular target, how the progression of the disease is, whether going to reduce or increase, or what is happening. So validations of target is important. You have to validate these targets. Then after target identification and validation, next step is you should have some molecules which will act on that particular target. As I told, whether your molecule, depending upon the target, whether you need a molecule which will activate that target, so it will helpful for that particular disease, or whether you have to be block the activity of that target, or you need partial agonistic or partial antagonistic activity. So it depends upon the particular target. So you have to identify a lead molecules, and after identification of lead molecule, you might be in early drug discovery phase. We are identifying a lot of lead molecule, maybe 1 lakh lead molecules. Then next step is you have to optimize these lead molecules. Maybe that lead molecule you have to be do QSAR studies. Then you have to identify out of these 1 lakh molecule which lead molecule is best one. So you have to be look for their toxicity profile as well as the efficacy profile. By using in early drug discovery phases, computational techniques are widely used, particularly these high performance computer facilities are used to simulate the environments as that of the body, and to check the interactions of that particular molecules towards the active site of particular receptors. QSAR studies are widely used to predict the biological activity of unknown compounds. So these techniques are useful without doing any experiments. to optimize the lead molecules. And after optimization of lead molecules, you have to select the best candidate, and you have to make a decision whether you have to be go for a drug development process or not. Drug development process is very much costly, so you have to make a wise decisions. Sponsors will take the decision, yes, they are interested to development of that particular molecules or not. And in drug development, first thing you have to do a preclinical trial. Preclinical trial means you have to test that molecules under in vitro or in vivo conditions. In vitro as well as in vivo conditions, you have to be check the that molecules or new chemical entities for its safety as well as efficacy profile. So both profile you have to look for. So in vitro conditions, you if you would like to check the efficacy of that new chemical entity, so you have to be test that molecule on a particular cell line of that particular disease. So you can get, yes, this molecule is effective for that particular cell lines or not. So next thing is you have to be see at what concentration that molecule is showing the efficacy, and for at that concentrations whether your molecule is safe or not. So for the safety purpose, you have to be perform your experiments, in vitro experiments on the normal cell line, not the diseased cell line. So you have to be take the normal cell line whether your molecule is safe against normal cell lines or not. And after completions of in vitro studies, you have to be select the molecules which you go for a in vivo studies. And in vivo studies, you have to use the animals. You have to be check the efficacy and safety of that particular molecules in particular type of animal. So as per guidelines, it's recommended that if you are using first time new chemical entities animals, then you have to be use smallest animals. Like you have to test your molecules in a mice, then rat, then guinea pigs, then monkeys. Like that, you have to be choose the animals for the testing of new chemical entity. It's not like that you have picked up the new chemical entities and tested in a monkey first instead of testing on rat or mice. In rat or mice also, first you have to be test your molecules in mice, then rat, then guinea pig, then rabbit, like that. So after that, if you are if that molecule is showing good efficacy as well as safety profile, then you have the sponsor has to be take the decision whether they have to be start a clinical trials, means whether they have to be conduct the clinical trial in humans or not. So all these informations related to safety and efficacy, whatever has been done in the animal, and these studies you have to be do as per the regulatory guidelines. So after that, you have to submit all these informations in a IND applications to start a clinical trial. That is the investigation new drug application. You have to submit to the regulatory authority of that respective country, and if regulatory authority approve that your IND application, then you can start a clinical trial. Means you can test your molecules in a humans, and in clinical trial there are different phases. First, you have to be do the phase one trial where you have test that new chemical entities in a healthy human volunteer. Our first objective is to check the safety of that molecule. We don't know how that molecule going to behave in a human. So first thing is we have to take the healthy volunteer, and we just a major objective is to check the safety in phase one trial. And after, if this molecule is safe, or you have to do calculation of maximum tolerated dose, and then you have to be planned for a phase two trial. That is the in that phase two trial you have to be recruit the particular disease patients for which you have developed that you are going to develop that particular molecule. Suppose you are going to develop a molecule for diabetes, so you have to take a diabetic patient. So in phase two clinical trial, our inclusion and exclusion criteria is very strict, because we don't know how this molecule, okay, this molecule is safe in healthy volunteer, but we don't know whether that molecule is safe in the disease conditions or not. So in phase two, we have to do the proof of concept study, whether that molecule is safe in particular disease conditions or not. Then you have to be go for a phase three clinical trial where you have to lose the inclusion and exclusion criteria, and then you can take a patients who have diabetes along with hypertension or any other thing. So that phase one, phase two, phase three, all clinical trial data as well as animal data you have to put in a NDA application, that is we call new drug application, and submit to the regulatory authorities. And if regulatory authority approve your NDA application, then you can market that particular molecules in the market. So after marketing also you have to be conduct the post marketing studies where you have to be submit to the regulatory authorities at regular interval of time about the safety of that molecules in a large group of population, that is we call pharmacovigilance studies. So all these things we have to do, and this is the journey of new chemical entities, and depending upon post marketing studies, many of you know many molecule has been withdrawn from the market, many molecules has been withdrawn by the regulatory authorities due to their safety profiles. So these are all, I just give you the overview about drug discovery and development process. So one by one, I will discuss how these computational techniques are useful in the drug discovery process in the next lectures. Thank you very much. I hope you understand actually about the overview what actually the drug discovery and development process is. Thank you.